Okay. Listen. I think if I give off energy, I give off the energy that I could easily spend this whole period discussing musical theater. But I'm not going to. Okay. So quiet you. All right? I keep that in mind. To what? Okay, no. It was a nice try. It was very good. We can discuss our Sondheims and our Lin-Manuel Mirandas at a later date, but right now we're going to get going. So, just a quick reminder, here we have, stop reacting to my things while I teach, Mr. Calford. So here we have the chloroplast. All right. You probably don't need to label that, seeing as it's, labeled there, but here we go. Here we have the chloroplast, which is another one of these double membrane structures, right? So we have the inner membrane and we have the outer membrane, but the inner membrane folds up into these interesting structures. All right, and the thylakoid membrane folds, and they'll refer to each of these individual folds sometimes as a thylakoid, but they call each stack of them a granum. So there will be multiple stacks of them called grana. Good. Now we need to get just a little bit more language. And the last two pieces of language are that we end up with this space around the grana inside the inner membrane, which is called the stroma. And then we have the space inside the thylakoids themselves, inside the grana themselves, which we refer to as the lumen. Okay. So, in a way, this has one more space to it than does the mitochondrion. Does that make sense? Because there is still an intermembrane space, though I've never really seen the intermembrane space as be described as a functional part of this, but it does exist, right? We have a space between the inner membrane and the outer membrane. And then we have what in a mitochondrion would just sort of be the matrix. We have yet more kind of space there because there's these foldings inside of there, the thylakoids that form these grana, and the granum together have both an interior space, which we call the lumen, and the space that surrounds them that we call the stroma. So the language gets to be a lot. So we're just going to write some definitions here before we go so we can be sure when we use those words we know what we're talking about. So for the stroma, we're going to just describe this, and this is not a very technical definition, all right? This is going to be a very just kind of plain spoken. So don't then judge me compared to like a textbook definition. But we'll call this inside the space inside the inner membrane. I'll say of the chloroplast. Now, whatever, we know we're talking about the chloroplast. But outside of the thylakoids. All right, and we'll call the thylakoids individual disc-shaped structures inside the stroma. So individual disc-shape membrane-bound structures found in the stroma. All right, so we have the stroma, which is the space inside the inner membrane. Inside of those, we find the thylakoids. Inside of each thylakoid, there's a space filled with fluid. We refer to that space as the... Great. So this is the space inside each thylakoid. Collectively, it's the space inside all of the thylakoids. thylakoid is one of those words that like sounds like a bad name for an alien in like a low-rent 50s sci-fi movie. Doesn't it? Doesn't it kind of have that energy? Like a science fiction writer came up with that word. Anyway, that's the end of my thought. And then granum, grana. Each granum is a stacked group of thylakoids. And again, granum is singular, but keep in mind it's a singular stack, right? So a granum consists of several thylakoids. You guys have quiet energy today, and I find when you have quiet energy, I feel less awkward if I also sit. Does that work? When a class is raucous, I feel like I need to stand and sort of conduct. And when a class feels too mellow, me standing up there feels like I don't know, I feel awkward. Anyway, yeah. It almost doesn't matter in as much as like this, I mean, okay, don't draw this, but you can imagine what you're seeing here is sort of cross-sectional, so you can imagine this as being that layer of phospholipids. So that itself would be the inner membrane, and then that would be like the surface of the inner membrane. Did that answer your question? Cool. An intermembrane space full of intermembrane fluid. And again, unlike in the mitochondrion, the intermembrane space of the chloroplast isn't where any of the action that I'm aware of happens. I'm sure there's things that happen in that space, but the way it happens to have evolved, the intermembrane space of the chloroplast is not the place where sort of the bulk of the reaction happens. It happens in the grana.
Kind of weird to think about how like the insides of you are just very wet, but you don't feel it. That makes me extreme.
I'd like to move on. Changing gears. That was such a Monday morning, like grade 9 boy comment. That was a comment begging for a word technically being thrown in there somewhere. You know, given how wet the insides of your cells are, technically you're wet all the time. Anyway. Anyway, okay. Does this look? Does this look familiar? Yeah, it looks a lot like the protein complexes that we see in the electron transport chain, doesn't it?
Wait, so all this is in that little thin purple layer that you like drew?
No. Oh, well, yes, except, all of this is embedded in the membrane, but specifically, it is in the membrane between the stroma and the lumen. So where is all of this happening? We'll actually draw it here. And in fact, if you look at my notes, I've included a helpful little drawing that I'm going to draw. See, right there. Because I rehearsed these. Believe it or not, this is me rehearsed, team. Okay, so where is this happening? This is happening, we are being told, between the stroma and the lumen. So if we look at a very schematic chloroplast here, we have the outer membrane, we have the inner membrane, and then inside of the inner membrane, we have the grana, and each granum is made up of a stack of thylakoids, right? Kind of like that, right? So this here, this section of membrane would be, say, there. Does that kind of make sense? I don't like how that looks, so I'm going to redraw it. But that's the idea, is that all of this would just be one section of the membrane that separates the thylakoid lumen from the stroma. All right, so that would be like here is this, and boom, we're going to blow it up, and this is what we're looking at. That make sense?
So, like, all this is in that little thin layer.
Yeah, that's right. It's embedded in the membrane. So, and so we have the stroma. Is the stroma the section out here or the section in there? Which one of those is the stroma? That's right. So this is the stroma. All right, so that would be the stroma. And if we wanted to, we could, I'm not going to, but if we wanted to, we could kind of say like that lines up with all of this section here, right? You know what? Oh, I don't hate that actually. Obviously, you're not going to do that, but for me, that actually isn't so bad. And then what do we call the space inside? The lumen, and I'm going to do that in a blue.
So those are the discs, and then the blue is the inside of the discs.
That's right.
Okay.
Which we're referring to as the lumen of the thylakoid, or the thylakoid lumen. So stroma is the fluid surrounding each one, and the lumen is the space inside each thylakoid. And that doesn't wash that out either on here, does it? Enough comments out of you. No, it's good. I liked it. Okay. So does that give us a sense of the space we're looking at? This mechanism should look awfully familiar. Does this mechanism look kind of familiar? Yeah. Right? Yeah. Well, and there's not really a specific opposite direction in as much as like there's no, but you're right in that in the electron transport chain, it is in the sort of the innermost sanctum. It's in the matrix that the H plus ions are going to return to, and they're going to make an ATP. But if you look here, this should look familiar because it's sort of a recycled design, right? What are we making here? We're going to make some high energy molecules, specifically in the form of we're going to charge up, we're going to reduce via redox reaction some ATP, and we're going to meet a new friend who is closely related to an old friend. What do we have here? I would like to never say NADP again, all things being equal, but NADP becoming NADPH, right? Which obviously looks very similar to NAD becoming NADH, right? It's a very similar molecule. We'll show you the difference between them. So this is another kind of form of a little electron transport chain. We don't call this the electron transport chain, but this is what we're going to look at today. This is the light-dependent reactions. So if you look here, the first element we come across is called photosystem 2. Now listen. Is it another classic science thing that the first thing that happens is we enter photosystem 2, and later on we're going to find photosystem 1? Yeah, guys, I don't name stuff. We discovered it second, all right? Turns out it's the first one that in line. I don't make the rules. So the first element is called photosystem 2. And the other thing we sometimes call it, and I think this is a better name, or P680. We'll talk about what that name means in a minute. Well, we'll write about what it means in a minute, but I'll put it here. Its ideal absorption is 680 nanometers. That's the area of light that this particular protein complex absorbs best. It will absorb along that whole spectrum we saw yesterday, but its peak absorption happens at 680, which is actually relatively low energy red light, but that's what it can absorb most. So P680 will absorb a photon of light. Remind me what a photon of light is? Yeah. It's a single quantized unit of light, so we call that a particle of light. And it uses its energy to oxidize water wherein water is going to break into half of an oxygen molecule. So if this happens a couple times, we'll get a whole oxygen molecule. And it's going to release 2H plus ions. And since it didn't used to have a charge, if it's got two positive charges now, what else is it going to release? 2 electrons. So we call this an oxidation reaction because it's losing 2 electrons, right? So photosystem 2 is going to be reduced because these are going to enter photosystem 2 slash P680, whatever you want to refer to it as. I'm going to typically refer to it as both, so you get used to both names because you will definitely see both names. All right. So the electrons are transferred, just like in the electron transport chain, through a series of redox reactions. The 2 R's in transferred, there totally are, aren't there? Oh, that looks worse to me now. 2 R's? Yeah? Okay. Okay, and they're going to go through this process here. They're going to be transferred from photosystem 2, which again we also sometimes call P680, to a molecule called plastoquinone, or the plastoquinone complex. Now, again, you don't need to know the specifics of this, but plastoquinone, where is it found? Inside the matrix, or not inside the matrix, sorry, inside the phospholipid bilayer, specifically in the actual lipid portion of it. Would it be hydrophilic or hydrophobic? It's inside, right? Look, it's right there. So it's exposed to all these fatty acid chains, so it's got to be highly hydrophobic, right? It's a protein complex, so you could probably go look at that protein amino acid chart and figure out which amino acids will be on the surface of this, right? They're going to be highly hydrophobic ones. But if you look, it takes on those electrons, and just like in the electron transport chain, when it doesn't have the electrons, it's attracted to photosystem 2, but it changes conformations. Once it has the electrons, it's no longer attracted to photosystem 2, but now it will bind to the next unit, which is called the cytochrome B6F subunit. Cytochrome B6F. Good name for it, right? If you wanted to use that for your next dog, you could. You could call them like Cyte for short. I don't know. I think if you went to the dog and somebody said, "Who's your dog?" and you said, "This is cytochrome B6/F," they'd be like, "Oh yeah, he's a cute puppy." So from plastoquinone, it gets passed to cytochrome B6F. From there, it gets passed to, actually, we'll pause there for a second, because what happens here? It pumps a H plus ion into the lumen. By the way, when water broke down, it also released H plus ions into the lumen. Ignore the numbers in this chart, by the way. It just shows everything as 1, but we know that actually this releases 2 electrons. This releases 2 H pluses, and actually 2 H pluses get pumped here, but we don't need to worry about the number. That's okay. we'll just say that cytochrome B6F, which releases H plus ions into the lumen. Sorry? Nope, this is showing the H plus coming in from the stroma, which is the next thing we're going to say. Yep.
So it's the B6F that's pumping the H plus ion or between plastoquinone and cytochrome?
Good question. So the plastoquinone took the H plus ion from the stroma. That's the next thing we're going to write, actually. This H plus ion was carried to cytochrome B6F. By PQ plastoquinone from the stroma. It'll take some time to remember the details of where everything comes from, but the big thing to take away here is that we've seen this game before. What are we doing? We are creating a situation where we have an electrochemical gradient. We have a low concentration of H plus ions on this side of the membrane, and on this side we have high concentrations of H plus. Does that make sense? When we do that, what do the H plus ions want to do? They want to equilibrate. They want to equilibrate, right? They want to, and when I say want to, of course, it's not like they sit down and have a committee meeting and say like issue one, and some of them take minutes and some of them take notes and they form a subcommittee. Rather, it's just the natural tendency of things to move from high concentration to low concentration. So they want to move back to low, they tend to move back to low concentration. It is also the tendency of charge to separate. So having a high concentration of H plus ions means that they're going to repel each other, all of which means there's going to be what we call electrostatic pressure or a chemiosmotic pressure for the H plus ions to move from where they are accumulating in the lumen back into the stroma. So the electrons are passed on so the E negatives move from B6F, cytochrome B6F, into this next molecule that, just like last time, remember, we had one that moved inside the membrane and one that moved on the surface of it. Once again here, this molecule, plastocyanin, moves along the surface of the membrane. It would be hydrophilic or hydrophobic? Hydrophilic, right? Because it's not exposed to the lipid insides. It's opposed to it's attracted to the phosphate heads of the bilayer and to the water inside the lumen, right? So plastocyanin is going to carry those. I guess we'll write it out once, and then usually we just use the PC. Quiet you. Is that better? No problem. Plastocyanin. These things happen. And then to what we call photosystem 1. Photosystem 1, which we also sometimes call P700, because its peak absorption, though again, not the only wavelength that absorbs that at all, but its peak absorption happens at 700 nanometers, which is a little bit lower energy. And PS1 absorbs another photon, re-energizing the electrons. Then the electrons get passed to ferredoxin, which we usually will just show as F or FD. I think they showed as FD on this particular diagram. So they get passed to ferredoxin. And then from there to FNR. Ferredoxin something reductase. Ferredoxin NADP reductase. Because it takes the electrons from ferredoxin and passes them to NADP, reducing it. Yeah, so it's ferredoxin NADP reductase to FNR, which I'm not going to write out. But it's here, which uses the energy and which passes them to, well, which we'll just say, which catalyzes the reduction. NADP plus H plus into NADPH. This does two things. It's the resting place for those electrons, and it uses up yet another H plus in the stroma. So this is the resting place for the electrons. And uses yet more H plus in the stroma. So that further lowers the concentration of H plus in the stroma, which means it's going to heighten even more the difference in concentration between them. Yes. Take a minute to write that. I'll shut up for a minute while you write it. Yeah, but not. Yep. Yep. So repeat the question. Sorry, I didn't hear it.
So the part where the plastic ion moves to the cytochrome B6F, why is more H plus in the already has?
Because this is just like the electron transport chain is a machine designed to build an electrochemical gradient so that that electrochemical gradient can be used to drive protons through the channel with ATP reductase to store energy. We're doing the same thing here. So that molecule is designed to take H plus from where it already is in low supply and move it to where it's already in high supply. So plastoquinone and cytochrome B6F together are pushing H plus. They're basically acting as a pump, but it's not a single unit pump. So we draw it a little bit differently. It's a two-step pumping process. So that H plus ion binds to plastoquinone, which then transfers it to cytochrome B6F, which releases it into the lumen. But functionally what we're doing is we are making this gradient. We're building this gradient. And the reason we're going to build this gradient is so that we can use our old friend, ATP reductase. Or synthase, rather. Does that make sense? So, and the reason we're doing that is that we want to make some ATP. Now, in this case, this isn't actually ATP for the cell's use, which is why we don't quite call this like the powerhouse of the plant cell. This ATP is going to be used inside the chloroplast to build sugar. But at the same time, we need some energy. We're also storing energy in the form of NADPH, which just like NADH is another one of these high energy storage molecules. And it's roughly equivalent to NADH. Basically, this evolved alongside it, or it evolved from the mitochondrion, or the mitochondrion evolved from this. We're not entirely sure which, I don't think. But this molecule serves a similar function to NADH. Did that answer your question at all, actually? Hey, on a Monday. Look at that. That might be the only thing I do right today. So I'm going to take it, and I'm going to be pleased with it. All right. Then the last thing we're going to say is all of this creates an electrochemical gradient. And ATP synthase channels allow H plus to move back into the lumen, right? Sorry, not to move back into the lumen. They're already in the lumen. To move back into the stroma. Catalyzing ADP plus a phosphate group that's free into ATP. And these are both our anabolic reactions that store energy. All right? Okay, so the advantage to this being quite similar to the electron transport chain is it's another good reminder that like cells have evolved and modified over time. So we see a very similar structure here to what we see in the mitochondrion because they're related to each other. They have evolved side by side of one another. So it makes sense that they would use similar structures, right? It's also advantageous because if you understand that you can pump H plus ions, separate them with a membrane, and then put this thing called ATP synthase here to allow them to flow back where they want to go and use that to power making ATP. Well, if you understand it in one place, you also understand it in the other place. So that's all good. The one negative, is it easy to kind of confuse these two things? Some of the details? Definitely, absolutely. But that's okay. We'll have time to study and practice and make sure we keep the details clear. But do you understand the basic broad stroke here, which is in Krebs cycle leading into the electron transport chain, we do this same thing to make ATP, and the power is coming from NADH and FADH molecules that came from the sugars, right? Here, where is the energy coming from? We're not burning a sugar here. We're not breaking down a chemical. Photons of light. Photons of light are providing the energy in this place. And how many times in this process do we absorb light and get energy put back into the system? Twice. Once at photosystem 2, interestingly the first time, and then later on in the system at photosystem 1. Does that make sense? Yes. We call these P680 and P700 because if you remember, different pigments have slightly different peak absorptions, and yeah, they're actually both chlorophyll A molecules. Chlorophyll A normally has a strong peak at about 680, and in P700, chlorophyll A has actually been slightly modified, and it has a peak at P700. Though to be clear, that is by no means the only place it absorbs light. There's also always big absorption down here in the blue spectrum. Does that make sense? But we kind of differentiate. In photosystem 2, chlorophyll A has this conformational shift, and it absorbs light a little bit lower energy than it otherwise would. You don't need to know that, but just if you're wondering where the names come from. Does that make sense? Turns out if you slightly change the shape of a pigment, the colors of light it absorbs and reflects change slightly. And when you put a pigment in a big protein complex, they have their shape change slightly, and that can affect their absorption spectra some. Okay. So, the end game here is basically that the plant has taken sunlight and turned its energy into an NADPH molecule and an ATP molecule. And basically, for every 2 photons of light we absorb, we get an NADPH and we get an ATP. All right? We're not going to worry about the specific numbers in this case because there's all kinds of things that factor into the efficiency of this process. And it's not kind of the main concern. But the basic principle is we absorb photons of light, we produce NADPH, and we produce ATP. Is that fair? Does that feel okay? All right. Again, the purpose of those molecules is that they contain energy. They hold energy for us. If you look here, if we are measuring the energy of the electron, really what we're measuring is the free energy. In other words, the energy available to work in the electrons. And when the whole things start, the electrons don't have much in the way of energy. Now this one here does show you the basic numbers, which basically say that for every 4 photons of light, why are they choosing 4 photons as that metric? Well, on the previous page, do you remember that we said that photon of light, and really it was 2 photons of light, gave us half an oxygen? Can we all agree that that's an obnoxious unit to work in, half of oxygen? So basically, for every 4 photons, we will end up being able to release 4 electrons, right? Which is enough to produce 4 H plus ions, and notably, it's enough to make 1 oxygen molecule. So that's why we've factored this whole thing up and scaled it up by 4. Does that make sense? Yeah.
So like a single cell of this could only at a moment in time make or take in 2 photons. Like they'll never take 1 photon?
An individual pigment will actually only absorb 1 photon at a time. Okay. And so it won't be until 4 photons have been absorbed that we would think of P680 as sort of having done 1 cycle or 1 round of this reaction.
So what we just went over was half a cycle?
Basically.
Okay.
Is that fair?
Yeah.
Cool. So as 4 photons of light get absorbed, it's possible that they could all be absorbed by the central reaction site. But remember we said that the photosystem is actually made up of a bunch of pigments, and more often than not, they have to kind of play this leapfrog game until they get to the central one. When they get to the central one, that's when the reaction actually happens. And when the reaction actually happens, that's when the electrons get energized, and now they're full of energy, right? From there, they get transferred, if you recall, to plastoquinone. Right? And then from plastoquinone, they get passed to the cytochrome complex, right? And here it says that ATP is produced. Does cytochrome directly produce ATP? No, what cytochrome actually does is it pumps H plus ions, right? And it is the pumping of those H plus ions by cytochrome that will get ATP made, but it's not the cytochrome that actually makes the ATP. What's the molecule that actually makes the ATP? ATP synthase, yeah.
So it's releasing oxygen at the beginning of this reaction. Is that like the oxygen that's like released to the air?
Yep, exactly right.
And then what's like, so like, and then I assume during the Krebs cycle, the CO2 is what we
Yep.
breathe out. What kind of like path does the CO2 take so we actually breathe it out?
Oh, great question. So as it builds up in your cells, we have another chemical gradient, and we know that things like to spread out. So it's going to try to get out of your cells, which it will do. And in your body, what surrounds all of your cells is your blood. So it will make its way from the cells into the interstitial fluid. From the interstitial fluid, it will find its way to capillaries, where it will enter your blood. And then inside your blood, it'll get carried. And then it'll make its way to it'll make its way to the heart. But in the heart, there's no real place for it to go. In the veins, there's no real place for it to go. In the arteries, there's no real place for it to go. But the heart will eventually pump it to the lungs. And in the lungs, there's this massive network of capillaries again, this time surrounding an air sac where concentrations of CO2 are low. And then they'll move out of, just naturally. There's no mechanism that pumps them out. Just things like to flow from where there's a lot of them. So there's a lot of CO2 in the blood, and it will just naturally flow into the alveoli, the air sacs of the lungs, where there's not a lot of CO2, and we breathe it out. And because we're constantly breathing it out and breathing fresh air in, the alveoli are constantly drawing CO2 out of the blood into them. Did that answer your question? And then very similarly, this is happening in the leaves of plants. The leaves of plants are very, very, very thin. So as oxygen concentrations grow in the cells, they'll make their way out of the cells. And right next to the cells of the leaf is just the air. And so they will just naturally flow into the air straight out of the cells. They can just interface directly. They'd also make their way into the fluid that surrounds all the cells of the plants, which, if you recall, transport in plants, that's going to end at the stomates, right? And will evaporate up into the air. And so that's just a direct interface. Did that answer your question? That was a good question. So yeah, this is where H pluses get pumped, and then that's used to make ATP. Just so we're not thinking that cytochrome complex itself makes ATP. But the cytochrome complex will pass its electrons on to plastocyanin. Now, this whole time, we're going through this second electron transport chain that we've learned about, this electron transport chain being the one of the light reactions. The energy in the electrons is going down. Well, that's because they're doing work as they go, right? They're getting passed around, they're changing conformations, they're pumping H plus ions, they're losing energy. Once they get to photosystem 1, more photons get absorbed, and since more photons get absorbed, the energy level gets higher. And then, as we move down the electron transport chain here, those electrons will get passed on to ferredoxin. Ferredoxin will be activated by the ferredoxin NADP reductase subunit, which will take NADP, and it will take H plus ions, and it will smack them together to form NADPH. And remember, this stores energy, and this stores energy. So we've taken sunlight that's full of energy, and we've got it now, that energy stored in stable compounds, ATP and NADPH. Yeah?
Other diagram, it says ATP is made up at this last one, and
Yep.
this diagram.
Yep, correct. So listen, excellent question. The cytochrome B6F pumps the H plus ions that are used by ATP synthase to actually make the ATP. So in the next diagram over, they're just trying to simplify things by not drawing both of those, because this actually happens outside of the chain. So cytochrome B6F is responsible for this in the same way that, like, God, what's an analogy? You know, like if you eat candy and it makes the energy level high, do you say the cookie's responsible, or do you say, like, you know, or do you say, like, your intestine's responsible for absorbing the energy? Well, you could kind of say both. Does that make sense? The cytochrome B6F is responsible for pumping the H pluses that we need to make ATP, but yeah, it actually happens over here. So this diagram over here is just trying to simplify that, but you're right. Oh, God. You're absolutely right that what it really does is it pumps the H plus ions, and those H plus ions are used to make ATP by ATP synthase, which is like this silly looking thing here, right?
So, the ATP originally supposed to be after the NADPH, or?
Well, the ATP isn't made as part of the electron transport chain, which is why it's not really shown on here directly. It's shown as something else. So if you look, the NADPH is made directly by the electron transport chain. In fact, that's where the electrons that started out in photosystem 2 end up. Whereas the ATP is made outside of the electron transport chain, but as a result of it. So this diagram here isn't showing anything outside of the electron transport chain. So it shows it at this step, because this step is the step of the electron transport chain of the photosystems that allows ATP to get made. Did that make sense? Are you just saying that to shut me up? Okay, good. All right. So we'll just remind ourselves here that the energy that ends up in ATP and NADPH by the way, if you're remembering NADPH versus NADH, it helps to remember that NADPH is only found in plants. So that's the one you find in the in photosynthesis. NADPH is found in photosynthesis. Does that help? Sure it is. Okay. Comes from photons. absorbed by PS2, which is also known as P680, and photosystem 1, also known as P700. Good. The 680 and 700 of those names refers to each system's peak red light absorbance level. Refers to each molecule or each complex's peak red light absorbance wavelength. Measured in nanometers. And if you look, just like we've shown before, there is the NAD plus NADPH complex. It looks very, very similar to just NAD plus and NADH. It's got that same thing going on where it's got the positive charge here in its NAD plus form, and then here the double bonds have shifted. We add an extra hydrogen on, and now we refer to it as NADPH. I feel like I've already said this like 4 times, so I wasn't going to make another note where we say that this is how energy gets stored. Do we want to write a note that's saying this is similar to NAD to NADH in cellular respiration, but is a slightly different molecule? It is. Are we good? We're good? Okay. All right. Excellent. Just want to make sure we're clear. If you want to see the difference in structure, here's the difference in structure. So,
This is in photo, or this is in like human.
so this one here is used in cellular respiration. And this here is used in photosynthesis. What's the difference between the two? The only difference between the two is that this one has this phosphate group at the bottom, which is called, why we call it NADP. So we have nicotinamide adenine diphosphate and nicotinamine adenine. Yeah, the truth is that D actually refers to diphosphate, and then here there's yet another phosphate group, but that's okay. Okay, so the only note I'm going to make is that it makes sense that these are similar. Evolved with, from, and alongside the reactions of cellular respiration. Remember, like in grade 11 biology, first of all, remember grade 11 biology. Remember how easy that was? Remember when we were like, you have a stomach. What's your stomach doing? It's like it churns food. And now it's like, so NADPH is produced by, do you see what I mean? Like, of course, a lot more challenging than that course, but I think rewarding too. I hope you find it so. But remember when we talked evolutionarily about the idea that, like, you look at a bat's wings and it has all the same carpal bones and all the same metacarpal bones that your hand does. And you look at like a whale's flipper and so does it. And that's because even though those things don't have hands like we do, they evolved not from us, but they evolved alongside us from common ancestors. Well, likewise, the systems that end up as the chloroplast evolve with, from, and alongside the systems that end up as the mitochondrion. So we see recycled materials here, but they have also been evolutionarily branched off from each other long enough that we see differences, which makes sense because they serve different functions. Does that feel okay? Does this feel like a lot on a Monday to you? Yeah, me too. You guys are doing great. And can I say, you guys really are doing great. This is dense material, and studying biology can sometimes feel like a bit of an information dump / an information kick in the proverbial teeth. And you guys have been consistently handling that quite well. So just stick with it. It's the biggest thing I can say is I remember that I used to, in a few of my biology classes, we'd just have study groups, and we would honestly just get like 5 of us get together, and we'd order pizza and wings and be like, all right, you go. Describe photosynthesis. And somebody would just go. Be like, all right, what'd they miss? And we'd talk, like, just talking your way through it and just using the language a lot is going to help you sort of familiarize yourself with it. Does that make sense? And obviously, I'm a big believer, draw it out, write it out. There's nothing here that you need to know the structure of whatsoever. We don't care about that. So don't look up the 10,000 atom structure of like Faradox and be like, I'm going to memorize it. None of that matters, but understanding the principle is really important here. And so let's just take a quick look here and sort of draw some conclusions from it, which is what's going on here. And if we look, what's going on is that in the electron transport chain itself, what we're going to do is we're going to absorb light. First step, photosystem 2. That's going to provide enough energy to take a water molecule and break it apart, right? When we break that water molecule apart, the electrons from the water molecule, 2 of them, are going to enter photosystem 2. Now those electrons get energized. At the same time, we release the byproducts of that water, which are oxygen, good for us, we breathe it, and H+ ions, which is going to start this process of lifting the concentration of H+ in the thylakoid lumen, or what they call here the thylakoid space, which has this high H+ concentration. Those electrons are going to get passed on to plastoquinone. Plastoquinone actually uses that to absorb H+s from the stroma, what they're here calling the chloroplasm. Yet another word you'll see from time to time. You're welcome. It will pass the electrons and the hydrogen onto the cytochrome B6F complex, which will take those H+s and move them also into the lumen, into this inner space. Those electrons will get passed on to plastocyanin, which will pass them on to photosystem 1, where they will get more energy. They will get that energy from yet another photon being absorbed. At which point they will pass the now even higher energy electrons on to ferredoxin, which will pass them on to the NADP plus reductase complex, which will use those electrons to combine an NAD plus with NADP plus, with 2 H pluses, forming NADPH and leaving one of the H pluses behind. And then all those H pluses that have been pumped into the lumen are going to pass through an ATP synthase molecule, sometimes called ATPase, never called ATPase. And then it will use that driving energy to turn an ADP into ATP. And when all this is said and done, what we have is an NADPH molecule and an ATP molecule that can get passed on to the next step. And we're not going to worry about the next step today, but the next step is called The Kelvin cycle.
Like with
No, not Kelvin, but rather Calvin. Like
I could
like John Calvin, but not John Calvin, or like Calvin Klein, but not Calvin Klein. Sorry? Oh, what was it? They do vaguely look tooth-like, I suppose. So where to go from there? Do we want to write another quick summary? I think it never hurts because I think repetition breeds comprehension. So I want to write here, how do we write this in just brief words? Well, this whole thing is about capturing. Beautiful, perfect. It's all about capturing light, the purpose of which is to capture. Yeah, and you said the word to get there. Yeah, beautiful. So let's make that our first. Note. Light energy.
Wait, can I try to summarize this?
Please.
Okay, so the purpose of this
Mhm.
is to capture a photon of light to create enough energy to break down a water molecule. So there's an H plus ion, and that's about as far as I can get.
No, but that's really good, actually. Light energy is captured by the photosystems. That was really good, actually. It's really hard to do. Does anyone else want to try? We don't volunteer other people for things.
Did you hear what he said?
I did not.
He's like, you were really good. I can't beat that.
That was good. See that, Abdullah? That was smooth. Well done. That was both kind and generous. And who's gonna then be like, no, I sucked. You need to do that. That was good. That was well done. All right. I kind of like this approach. Light energy is captured by the photosystems and is used to do a few things. One, like Kiki told us, break water into oxygen, H plus, and electrons. I guess if we're sticking with numbers, and we could track each one. This goes into the air, and we breathe it. This is used to make what? Indirectly, but what are the H pluses used to make? ATP. Very good. And these enter the electron transport chain of the photosystems, right? All right. So then what else does the energy do? It pumps even more H plus from the what to the what, right? From the stroma into the lumen, right? Oh, if you found that you wrote these things way too big, just rewrite it. That's the long and the short of it. What's really happening here is light energy is captured by the photosystems, and it's used to break water into O2, 4H pluses, 4E negatives. The electrons enter the electron transport chain, which then are used to pump more H plus from the stroma into the lumen, and they're used to catalyze NADP plus into NADPH. And that's kind of it. That's what happens here, is basically we're using light energy to make ATP and to make NADPH. And the ATP and the NADPH are going to be used later on. Does that feel okay? Doesn't feel too bad, does it? All right. So what comes next? We're not going to learn it today. What we're going to learn tomorrow is that ATP and NADPH enter the light-independent reactions where sugar is actually made. Sorry? So do you mind speaking loudly as if I were an old man? The light-dependent reactions. We refer to that collectively as just the light-dependent reactions, and we refer to the next stage as the light-independent reactions. Good question. Ent. Like the large tree-like creatures from Tolkien's Middle-earth. That was helpful, right? Yes?
My brother's birthday is November 20th, and...
Okay, I'll put it on my calendar.
Oh, no, there's a point to this.
Okay.
His birthday cake is a Lord of the Rings themed birthday cake, and it's like a book, and it's opened up.
I might need to steal that.
And, yeah, his fiance, like, already picked it tonight. It looks really cool. His whole 30th birthday party is planned to be Lord of the Rings themed to this movie series.
It's amazing. I used to read The Lord of the Rings every year over Christmas, and I haven't done that in years. Now my daughter and I always watch the films each year, and Greg usually does too. But this year I want to read them again because it's been a few years for me, but they're wonderful. I will say if you plan to read The Lord of the Rings, they're wonderful and rewarding and very worth your time. But I will say there are also long segments where it's like describing walking in the woods and long elvish songs. But don't let that deter you. They're wonderful. They reward the reader. Cool, that's good. I haven't decided yet, and it depends on whether some materials I've ordered arrive today or not. We are winding near the end of this unit. We actually have at least one, possibly two labs we're still going to do. I like to try to get the material out of the way quickly so that you have time to review it and study it before test time comes, if that makes sense. But I also don't want to make this punishing. Do we think we're good for another lesson tomorrow and put this to bed? Or do we feel like we need a break in the middle?
No, we'll bed tomorrow.
Yeah? Okay. Then we'll do that tomorrow.